Thank you very much. This is the University Hospital in Vienna here. Very modern building. This is an old castle here. So we have some collaboration with the clinics, and sometimes we are contacted from cardiologists, and they want to know the reason for the disease. Now they have a suggestion that there's a sodium channel mutation, and we are screening that, and we are analyzing them the function in vitro. The problem we often have is that we, regarding this title, clinical utility of basic research, is the fact that our experimental results cannot explain the phenotype. So there's a significant disassociation between genotype and phenotype. So what we are doing is when we see such a ECG, we are doing the genetic test. Then we find a mutation, let's say in a sodium channel, and then we are expressing this mutated channel in oocyte system and in HEK cells. We're analyzing then the currents by patch clamp analysis, and then we are happy if we can see a difference between the wild type channel and, let's say, the mutant channel. And then, of course, we can tell the cardiologist, okay, that is probably the reason for the disease. But then the cardiologist sometimes may answer, okay, we have family members that are healthy, they have the same mutation, so there's a clear disassociation. And on the other side, we sometimes cannot see any defect, so the mutant channel shows the same features as the wild type channel. So that means that we missed something during the in vitro analysis, or this alteration here is just a natural occurring variation in the sequence. So we don't know actually. So there are now many factors known that can influence this genotype-phenotype correlation, leading to such a disassociation. One thing is there are metabolic factors, for instance the pH value. It has been shown that acidic cells produce a stronger or larger persistent current. There are a lot of environmental factors like xenobiotics and drugs that can affect that. That's all what we are missing in the in vitro system, of course. There are other genetic factors, and we have heard a lot of that during these 2 days. For instance, gender or aging. So one thing, this is very interesting, there are probably secondary effects by the long-term expression of a defective mutant channel. This has been shown in transgenic mice expressing a mutant channel, and they have shown an altered expression pattern for 33 other genes. So the mutation, the mutant sodium channel caused very different expression pattern in the heart. So if this affects then the disease, it's unknown for humans, but it's a very interesting result. Then inheritability is one point. In many of those cases, there's only one patient. So the number of patients is one. In many LQT3 and other diseases. So we cannot say it's inherited channelopathy because if we analyze the function, or if we analyze then in vitro this mutation, it can be, we may find gain of function, for instance, but in the same family there could be a patient with a Brugada syndrome. So there are mutations known causing Brugada syndrome in one patient, in the other family member, long PD. So if the patient number is only one, so we don't know. I will show you some recent data from our lab that the cell system, the expression system, can affect our results. And the second I would like to talk about is NAV1.5 splicing. We're always talking about mutations, but the sodium channel in the heart is also spliced, and there are functional splice variants in the heart. Here you can see again the topology of the sodium channel and the location of the different mutations. So we were very much interested in the N-terminus of the sodium channel because nothing is known about these mutations here, and nothing is actually known about the function of the sodium channel N-terminus. About the C-terminus we have a lot of data, but here what is the reason of the disease and what is the function of the sodium channel N-terminus? So we expressed these three mutant channels in 2 different expression systems. In the oocyte system, oocytes are incubated at 18 degrees, the core body temperature 37, so there's already a big difference. And we usually measure at room temperature. These cells are incubated at 37 degrees. Of course, that is normal, but measured at room temperature because you cannot easily measure at 37 degrees because the patch is not stable. Yeah? So we did this and compared then the results. So what we first found is that we got nice robust currents. That is normal. So we expected that, but we were very disappointed not to see a persistent current, because our hope was to see a persistent current when the N-terminus is mutated. So obviously the N-terminus of the sodium channel is not involved in inactivation. Here you can see a large persistent current in this in the first QT3 mutant channel. And when we did the same things, the same experiment in oocytes, so we got similar data. So there was no difference. When we analyzed the kinetics of those 3 mutant channels, we saw actually no change compared to the wild-type channel. In these 2 cases here, we only saw a larger window current in one of those mutations, so that can explain the phenotype. The recovery was enhanced also here in this, so we can explain the phenotype of long QT when analyzing kinetics in this case, but in the other 2 not. So we couldn't see any effect. In the oocyte system, we did the same study here, but there was no difference compared to wild type. So in the oocyte system, this was very frustrating for the doctoral student. Of course, we wanted to see an effect, but we didn't see any effect. We always used controls, so the measurements were good, but we couldn't see any effect. And mild effect only in this case here. Here you can see a sequence alignment of the sodium channel N-terminus. This is the starting methionine. This is the S1 of D1, so the first transmembrane helix. And in the oocyte system, so the currents and kinetics were similar or were the same as for the wild type. And we also couldn't see anything here in the mammalian system, only in this case, so we could unmask some defects, but not in the oocyte system. And you see, this is not a highly conserved residue here, it's a valine, but this is highly conserved here, so it's glycine everywhere. So it's very likely that this mutation indeed caused the disease, but because it seems to be an important amino acid here, but we couldn't see anything. So the mutations in the N-terminus did not increase the persistent current, and exchange of conserved residues did not alter the electrophysiological properties. And gain of function was only seen in HEK cells, not in the oocyte system. So then we did the same for the Brugada syndrome mutations at the N-terminus. There are several known, and actually we got very similar results. So we were very surprised to see a robust current in most of those mutant channels, except for this. There was clear loss of function, so no current. This can nicely explain Brugada syndrome, but you see in the oocyte system, there was a rescue of the current. So if you use the oocytes for expression, you cannot see this defect. So there's a big difference. And when we studied the kinetics, we only saw very mild effects in the HEK cell system, so almost no effect. So actually, we cannot really explain the phenotype. So Brugada syndrome. In the oocyte system, you see here, this position is also conserved. This position is also highly conserved. This valine is highly conserved, and probably this whole region is conserved and very important for sodium channel function because it's conserved and because if it is mutated, causes disease. So, but we cannot see it. No or only mild effects were seen even when conserved residues were affected, and if we detected loss of function in case of Brugada syndrome mutations, we had to use HEK cell system, not the oocyte system. Okay, so this is a summary of our results on the long QT3 and the Brugada syndrome mutations. As I told you, on oocyte system there was almost no effect. If we saw something, we had to use the mammalian system. So It turned out recently that the sodium channel N-terminus is probably involved in trafficking of the channel from the endoplasmic reticulum to the plasma membrane. So, and our hypothesis currently is that we will miss simply this information if we use a lower expression system. We have to use cardiac myocytes to study that, and not oocytes or HEX cells. And probably the N-terminus interacts with other cardiac proteins that are simply not present in those expression systems. So we cannot see any effect. And we have to use the appropriate expression system, cardiac myocytes or whole heart, to see the effect. The second point is alternative splicing that is often not considered in terms of genotype-phenotype correlations. There are several non-functional splice variants of NAV 1.5, especially at the C-terminus. So there's a good correlation now between heart failure and the increased expression of a non-functional splice variant in the heart. There are other non-functional splice variants, but there are no clinical data on that. But at the same time, there are at least 4 functional splice variants. So they indeed, the RNA is the same, then there's a splice, there's no mutation. And some of those splice variants show different kinetics compared to the wild type channel. So this splice variant was the first that was detected 10 years ago. There's just an exon missing here. Then another one is 1.5C. There's an additional glutamine at that position here due to splicing. Then in this splice variant, part of exon 17 is missing here, and this is a very interesting splice variant. Exon 6, we have 2 copies of exon 6 in our genome. So before birth, we use exon 6A. After birth, we use exon 6B. And the electrophysiological properties of both channels are different. And recently, this inclusion of exon 6A has been correlated or suggested to Brugada syndrome, or being correlated to Brugada syndrome, and it is possible that the exon 6A, if it is expressed in the heart, may cause loss of function. This has been shown for Steinert disease. Steinert disease is a muscle disease that the patients show abnormal ECGs, relatively similar to Brugada syndrome. And in Steinert disease, there's a kinase which carries multiple CTG triplets in the 3' non-coding region. This causes accumulation in the nucleus of a toxic RNA, and this causes alternative splicing in the heart. So that's why those patients die, because of heart problems. I was interested in this mutation here we found in Jena in a small family. This mutation here is located in the 4th domain. It introduces here this positive charge. It causes long QT3 and cardiac conduction disease in 1 patient. We have also a family tree. So we asked the question, can this mutation affect the kinetics of not only of the wild type channel, but also the kinetics when this channel is spliced, of the splice variant? Was there any difference? So if we have the splice variant in the hearts, then a mutation may alter a splice variant, but not wild type maybe. So, and first, here you can see the mutation, our result of the analysis of the kinetics of this mutation. It causes gain of function, so a left shift of the steady state activation curve, faster recovery, and slower inactivation at depolarized potentials, but also loss of function, so faster inactivation at less depolarized potentials, and the availability curve is shifted to the left. So we see in these patients long QT and cardiac conduction disease, and we can see also gain of function and loss of function in in vitro analysis. So when we introduce this mutation here into the background of NAV 1.5A or NAV 1.5C, we couldn't see any difference. So the mutation caused the same alteration in the background of wild type and in the background of the splice variant. Okay? So there was no difference. In case of this splice variant that we have before birth at least, the situation is a little bit different because the electrophysiological properties of the splice variant differ from the wild type channel. There's a shift of the steady state activation curve, so it activates a little bit later. Here this is the splice variant, but when we introduced the mutation, we again saw this left shift. So the relative difference was the same. So there was actually an expected effect. But this was not the case when we did the same, when we introduced the mutation in the background of this splice variant. Then this splice variant itself has very different properties compared to wild type. It shifts the curve to the right, but the mutation did not cause the left shift. The mutation, the splice variant itself caused a slower recovery from inactivation, and the mutation shifted this to the right, so recovery was even decelerated, so it was even worse. So this is my last slide here, summarizing these data on splicing. And here you can see the relative effect of the mutation on 1.5, so it shifted the activation curve, shifted the inactivation curve, and led to a faster recovery here. But in case of this splice variant, there was no effect here, and it was even a slower recovery from inactivation. If I superimpose all those curves, then you can see here what we have in the heart. If those channels are expressed in the heart, we have then various types of mutant channels and at least 2 or 3 types of wild type channels. So we have multiple variants in the heart, so it's not so easy. And yeah, that is maybe a starting point for a new project, but it shows how complicated it can be. So I would just like to say that alternative splicing should be considered when analyzing the effect of a mutation. Well, thank you very much, and these are the people who are involved. No change, no.
How do you explain that? What was methodologically different from what you do and what we done that show a lot of function in the channel?
So actually we don't know. I think the sodium channel, if for instance these mutations affect the binding to some specific cardiac proteins or something like that, if the trafficking is affected in the heart, and if the trafficking machinery is different in HEC cells, so we cannot see it. So the protein will be transported to the plasma membrane in HEC cells, but not in the cardiac myocytes. That's my explanation at the moment, but we don't know. Actually, it's very easy to see loss of function in oocytes and HEC cells. We don't know. But there is a difference between myocytes. I strongly believe that these are really patients, and it's really a good analysis. And the genetics is good, but we cannot see anything. So it has been shown for one mutation causing cardiomyopathy B1275N. I don't know, maybe you've read the paper. If you analyze this mutation in the in vitro system, you cannot see an effect, not a strong effect. And there was a long discussion, how can this mutation cause the disease? Then from the George group, there's a paper, a recent paper. They made transgenic mice expressing this mutation, and they showed the same phenotype as the patient.
One question. Do you think that some of the mutations, I think that the sodium channel is function of that channel is pathologically identical to one.
Yeah.
Do you notice specific sites that channel is pathologically has, and each mutation will act differently if you have PKA pathway acting?
Yeah, but at the end-terminus there's no PKA site. So, but at other sites, of course, yeah.
So if you combine pathways with that mutation, you know if it's different sites, and you have different text. You do it like the isomers.
We haven't done it. We haven't done it. But the phosphorylation sites are known.
Another question is, you mentioned that you modify the connection state of your channel. What do you think that will use like idea and will make sense in your research or
I didn't get it.
like if you modify the connection state of your channel, what do you think that will use as a many effects in your cells?
On the cell?
For instance, your capsule channel, right, has two stages, right?
Sodium channel, yeah.
So the one is open and inactive. The inactive stage is to protect the active stage, right?
Yeah.
So if that's not active anymore, it means that there's another active function that will occur, right? It's like in the middle of the first one, you would create an effect. So you think that modifications of that particular, I think it's the secret of the channel, would be more related to effect than
Yep. These sodium channel regions involved in inactivation are known. So this is mainly the D3, D4 linker. So and mutations there cause persistent current. Yeah, that's right. Yeah. But from the in vitro system, we cannot conclude anything regarding our. Sorry?
Last question.
No.
We're not able to do that mechanics, right?
No, not yet. No.
I have a question. You limited your studies to genetic mutation. What about epigenetics and a possible role for microRNA?
We have not done. Very interesting. I know your brother's doing this.